welcome everyone in the future, I suppose, to this panel from the Longevity Leaders Virtual Conference. We're going to be talking today for a little over half an hour or so about expanding age-related disease indications, basically how drugs that can target some of the fundamental biology of aging are being sent into the clinic today, and how they may eventually be used to treat different indications, including ultimately preventing us from getting sick in the future. My name is James Peyer. I run a company called Cambrian Biopharma that works in the space, and I'm joined by three excellent guests today. We have Bev Vaughan, Thomas Hughes, and Eric Morgen. I'm going to have you three, if you don't mind, each introducing yourselves briefly, and then we'll get into the panel discussion. Bev, if you would go first, then Thomas, then Eric.
Thank you very much, James, and hello to everybody in the future. My name is Bev Vaughan, and I'm the Programme Director of the UKSPINE, which is a connecting capability fund led by the University of Oxford across the United Kingdom, connecting academic hubs, not-for-profit and industry partners in delivering new therapeutics for multi-morbidity and increased health span.
Great. I'm Eric Morgen. My background is in medicine and computational biology. I'm co-founder and chief medical officer at BioAge Labs. We are a biotech company focusing on developing therapeutics for aging and age-related diseases. We have a platform to find the right targets for those diseases, and then we pursue in-house and also in-licensed therapeutics against those targets.
Hi, and I'm Tom Hughes. Please call me Tom. I'm CEO of a company called Navitor Pharmaceuticals. We're Cambridge-based, Massachusetts-based, privately held company focused on drugging the mTORC1 node as a targeting concept. Our programs span both activators of mTORC1 that regulate mTORC1 by way of sensor proteins for amino acids, to inhibitors of mTORC1, particularly selective inhibitors of mTORC1 that are being targeted for both chronic diseases of aging, but also with specific approaches to individual indications. Our scientific founder is David Sabatini from the Whitehead Institute at MIT, who I'm sure many of you are familiar with.
Yeah. Wonderful. Okay. Thanks everyone for joining me with this. We're going to have a discussion that hopefully will touch upon three of what I think are the key discussion points surrounding the story of companies in the aging space as it relates to how we are actually going to take drugs from academic labs, things that work in mice, and ultimately get them into people. The first is that we're going to spend a little bit of time understanding why this is such a big deal in the longevity world. The second is how small companies can start developing drugs that can extend the healthy lifespan or the health span of animals and get those into human trials for the first time. And third, I want to talk about how we can take a drug that looks like it's on the path to approval and start expanding out the utility of that drug to target health span generally. Multiple different conditions, big age-related conditions, et cetera. And so to kind of frame the discussion a little bit, as an investor in the space, one of the things that drew me into the longevity world a few years ago as this all started coming together was the notion that when you are extending the healthy lifespan of a mouse, say by inhibiting mTORC1, you're not just affecting this one thing called health span. The way that health span gets extended is by interfering with many different processes that are at the core of many different diseases. So in order to extend the healthy lifespan of a mouse, you have to prevent it from getting cancer. You have to prevent its muscles from wasting away. You have to prevent its brain from declining, all at the same time. And if we could do any one of those things to a human, it would be a fantastic, fantastic therapeutic intervention, right? If we could just prevent cancer or just keep our bodies non-fragile into old age. And here we had this evidence where we were doing something that was covering all of those things in a mouse. But there's not a clinical pathway to target all of those things at the same time in a human today. And so if say, and I'm going to continue using you as an example here, Tom, if you have an mTORC1 inhibitor that can extend the healthy lifespan of a mouse like rapamycin can by 10%, 15% or more, there's this enormous question of where do you start? Do you start as an anti-cancer prevention drug? Do you start it as a neurodegenerative disease drug, or do you go somewhere else? And this aspect of choice creates a huge problem for many investors and scientists starting small companies in the area, because there are many choices that might be right, but there's probably one that's the most right, and there are also many choices that are wrong. And for especially academic groups coming into the field, it can be difficult to navigate the right one. And so I think it would be really fun to start this part of the discussion to hear from Eric. So at BioAge, Eric, you guys have reviewed a lot of different pathways that all target the biology of aging, and you've really had to sort through, to get the right targets to go after, and also which clinical indications to think about for those targets when you bring them in. Can you tell the audience a little bit about how you've interfaced with that process a little bit, and kind of how you think of this problem in the aging space generally?
Yeah, absolutely. So you've really, I think, articulated the challenge well, is that you've got this really compelling thing you're doing that you think you've got, a target, an asset that will extend healthy human life. That's amazing. But then what are the nuts and bolts of how you actually pursue that, and how you also have high impact in an area that sort of checks all the boxes, that's something that's practical, that's actually going to work. And this is something that, we think about a lot for a number of our early stage programs, but also, we've, just in-licensed our first clinical stage asset. And so we're really in the trenches now in terms of thinking about how we're going to move forward with that. But to your point, absolutely, we've spent, years now developing a platform where we have collected human biological samples, spanning many decades, or we've interfaced with biobanks that have done that. And we now have access to sort of proprietary, molecular data generated using modern methods that tracks these people over long periods of time, and tracks their health span and lifespan, as well as individual disease incidents and outcomes, and allows us to select the right targets that we think are really impactful across all these different aspects of healthy human aging. But then ultimately, it's exactly what you say. You have to pick a specific first indication, and then ideally a second indication, third, et cetera. So I think there's a number of different aspects of that you want to get to, but what's really the pathway? I mean, so ultimately, you're targeting a specific mechanism, and that mechanism has to be right for your first indication. So of course, you have to check that box. It has to be relevant to the specific indication you're going after. Your indication has to have unmet medical need, has to be really clear, has to be a commercial opportunity. So there's all these very specific boxes to check. It's also great if that first indication you choose, is and also feels like a proof of concept mechanism for aging broadly. So you want positive effects in that initial indication to have broader implications across aging. So for example, Restore Bio in this space, they thought about immune aging, and this idea that the first thing you can do is maybe improve vaccine response in the elderly, and that's really known to be impaired in the elderly. And then you can go on to maybe trying to prevent certain types of acute infections, and that is nonetheless sort of inspirational and an example of something that if you did that, if you succeeded in that first trial, what you've done is improve the function of the immune system generally, which has all sorts of really broad implications for aging. So those are the kinds of key things you want to do, and of course, the right indication is also something that you can pursue in an efficient way, especially when you have limited funds, which is the situation most people are in. And so you want something that you can attack with a relatively short, cheap trial to get you to compelling indications, compelling data and results. So a lot of what that focuses on, even though our goal is, as you say, some really compelling long-term benefits for people, expanding their healthy, functional, happy, lifespan and health span. Maybe the way to start is with these more acute indications where you can show something in a really short amount of time, but that's nonetheless inspirational for those ultimate goals. And biomarkers really kind of come into play here too. And there's a lot of, I think, really strong potential for, if we could come to a consensus on molecular biomarkers that were, we believed would tell us about those long-term outcomes, that would also be, have a huge amount of value in this space. So yeah, I think I'll stop there.
Yeah, I think you touched upon a few really great points there, some of which I want to make sure we bring up again later in the conversation. Especially, I think what will come up again later is the point that you just made about biomarkers as a key part of making the prophylactic or proactive treatment of aging or of age-related conditions happen. And also your example of Restore Bio, I think we want to come back to. As certainly one of the emblematic trials in the space, although it hasn't worked out that well.
Unfortunately.
So I want to jump now to Bev, if that's okay, as we're focused on, because one of the topics that we brought up here is this idea of how academics that are starting new programs in the longevity field or in the health span extension field are beginning to think about who they would partner with, how they would think about advancing their programs forward. Since you're kind of at the nexus of some of the collaborative work out of Oxford, I'm just wondering if you've seen some of these challenges being faced and addressed by folks that you're working with. And then I want to jump over to Tom after hearing from you.
Oh, absolutely. Thank you very much, James. So some of the points that you picked up on, Eric, are very pertinent to the academic groups. Particularly, I was making a few notes around the biomarkers piece, and you talked about the data sets that you're accessing. Many researchers are now actually being signposted and thinking quite hard about where that data is and how they understand sort of the population demographic before they rush forwards with what they see as an exciting and interesting field. And I think one of the most important things at that point is getting them access to the right people at the right time, not just in terms of accessing patient data, which is problematic in itself, and actually getting the data on the biomarkers is problematic itself for a lot of the academic groups who have to make lengthy applications to Genomics England, UK Biobank, equivalents elsewhere in the world, but to understand what they're going to do with that and what's going to be a sensible decision. And I think academics in this field, we've seen quite a range of things coming in. A lot of things are still very early where they are really looking at those underlying pathways, and they've got great ideas that might target senescence or autophagy that have got really, really interesting data. But they're a little bit lost. They're lost in how they take that forward and where the interest is going to be to pick it up. And part of what we try and do and we see with our groups is then connecting them not just to the right data, but also the end of the pipeline to the clinical question, and looking at, you could have a great idea, but how would you get that into first in man and later clinical trials? So I think there's a lot of great science going on, and some is very quick off the starting blocks. But it's not necessarily formulated in such a way that's going to be very easy for any of the chaps on this call or in the wider world to easily pick up on. So I think as the field really does take off, It's about really good signposting and things such as Eric's resource that he's mentioned, how we standardize this and look at not just biomarkers, but physical markers and ways that endpoints in trials can sort of be looked at more intelligently across indications, and then feed that backwards. So it's almost you have to go to the end and come back again. I hope that makes sense.
There we are. Absolutely. I think that's a good way of viewing a lot of this, is that in order to get where we want to be 10, 15 years from now, where we can be doing trials based on biomarker surrogate endpoints that can be preventing different diseases, we have to kind of start from there and work our way backwards, asking how do we take this drug that we have in hand right now that seems to do something in mice, or does something like induce autophagy in human cells, and how do we plot that there? I think, Tom, I want to toss the discussion over to you a little bit, because Navitor has been shepherding this mTORC1 inhibition program forward out of Sabatini's lab for a while now, and you guys have really been on the forefront of asking this question. How do you think about this problem of where you start from the clinical's perspective, and how that informs where you want to go in the long term?
Yeah, and this is really at the core of our sort of daily existence, for the simple reason being that we have chemical matter that we can begin to deploy, and it's time to really find that path and to make sure that it's tractable. When you think about mTORC1, it sort of comes back to what you were saying earlier about this abundance of choice, and we have for mTORC1 almost an embarrassment of riches, in the sense that there are thousands upon thousands of papers that have carefully studied the pathway, and that provide a lot of insight into where inhibition might be relevant, or even where activation could be relevant. And so, this is a sort of unique situation in the sense that you have this sort of very rich tapestry of information to lean on. And before I go on to mTORC1, I wanted to just mention there are examples that don't have that sort of luxury of the benefit of a lot of information. For example, when FGF21 came along, with its relationship to Clotho, this appeared very quickly on the scene to be a very, very interesting principle. And because there wasn't very much known about it, it looked really, really attractive. And over time, the data that were generated began to temper some of that knowledge. And whereas we believe that this is a modality of tremendous interest, it's one that sort of is still continuing to sort of collect its data in terms of what can be done, what should be done, what needs to be done with it. But coming back to mTORC1, our approach, we're sort of constantly bathed in this understanding that rapamycin and inhibition of mTORC1 or even S6K, which is downstream of it, have been shown over and over and over again to be principles that can lead to extended lifespan. And as you mentioned, there are a lot of benefits that go along with that. So our approach has been really to ask the question of where is the strongest link. And if you sort of think to the rubric that pharma applies to, the larger pharma companies apply to target validation and choosing programs to move forward, they rely a lot on genetic validation of their targets as a sort of a de-risking that can be applied to any therapeutic approach they take. And so where we're looking first is where are those conditions in which there's a clear link between genetic dysregulation of mTORC1 through any of its components and the disease that we're approaching, and to really then make sure that the molecule matches well to that particular tissue type or cell type, and to start with a very solid foundation of here's a disease in which there's a clear genetic link between the pathophysiology and dysregulation of the node. Here are sort of a series of endpoints that are both disease relevant and mTORC1 specific that we can use to select dose and to carry forward this program. And only then would we be interested in sort of taking on an indication to carry forward. Beyond that, one can think of the other endpoints that you might measure in trials as sort of fringe benefits. In other words, the fringe would be improvement in glycemic control, the improvement in vascular stiffness or immune senescence being reduced. But that requires that you look in patients who have those issues as well. And so I think this is where the sort of public-private partnership could really come to play a very important role, is that if one has de-risked a program or a compound to the point where one could run sort of semi-chronic type studies, let's say for 3 months, could one collaborate with maybe a university-based research group that has an annotated population for which there's known biomarker endpoints, and basically screen your compound to see how that impacts in those particular patient segments. But there's a lot of like in the weeds of getting there. You need to show that your PK isn't different because they're elderly. You have to look at drug-drug interactions if they're on multiple agents. So it's all of these things become challenging. But I think this is where that sort of private-public partnership could really help us as smaller companies to advance these therapies. And again, I'm sorry if I drifted off of the scope there a little bit, but that's just some things on the top of my head.
So I want to actually. Oh, Eric, were you going to?
Oh yeah, just
there?
yeah. There's just something Tom said that really resonated with me. I just wanted to reflect that very briefly, which is just this idea that there are some targets where we know a lot about them, and some targets where we don't, and that's something that we feel just really strongly because using with an untargeted approach where we're screening like the whole proteome, the whole transcriptome, et cetera, for things, we have really compelling targets that very little is known about, and they ultimately might be super impactful. It's the way you say Tom, but there's, I think less confidence around exactly how you go ahead with those versus something where, you find it and there's actually, a robust literature around that mechanism, what areas it's relevant in, what tissue types, what animal models, et cetera. And that's something, you can definitely use for triage and around this idea of, we feel so strongly about human data. And you brought that up a number of times, Tom, that, some kind of human data around the mechanism and its relevance to a specific disease indication is, really important. And we start with that with our own cohorts. Do we see that kind of signal around a specific disease? But then, exactly around the kinds of things you mentioned, you then go out into public data sets and figure out, what do we know about the transcriptomics across different tissues in mice and humans, et cetera. And then last, I think another really important point you brought up with the sort of partnerships is that, whatever data you start with, ultimately, if you think your mechanism is relevant to a specific disease, a really great thing to do is go and find a specialized cohort that already exists around that disease, that makes sense for testing your mechanism, and find samples and test those samples and see if the PD markers or biomarkers relevant to your mechanism are changed in that. So there's this sort of triaged approach you can use, and that's exactly the way we think about it too. So very interesting points, Tom. Thanks.
And just
if I might just jump in quickly to say, sorry, I heard someone say recently that mice lie and monkeys exaggerate, and that I think that keeping ourselves stitched into the human pathophysiology and human experiential data is really going to be critical if we're not going to, if we want to do this efficiently. That's just all I want to say.
Absolutely.
Sorry,
Bev, go ahead.
Sorry, getting my cues wrong again. Just to pick up on a really good point there about the public-private partnership and the less well-studied targets. And I think that's exactly where academic groups and consortia working with industry, which really come into play, that, where you have that riskier target, but you can have something like a group like our own taking that forward and studying that in early stage. And if there's validation that actually you've got something that's worth picking up and taking forward, it then goes out of the academic consortia back into your industry pipeline. And I think, it's that sort of risk sharing and also accessing those cohorts that sit within those consortia where we can work together so that one is feeding into the other and adding value and power of the data.
Excellent. Yeah, I think it's fascinating for me looking at this discussion because there are really sort of 3 stages that any drug in this field will move through in its ideal lifetime. There's the one that we've been discussing so far, which is how do we find the right drug, the right target, and the right first indication to go after. And then the second stage is how do we expand that drug to other indications, which is something that we haven't touched on too much, but I would love to hear the panel's insight on, is that if you get a drug approved, let's say for an orphan disease in its first indication, or for maybe a common thing like kidney disease, then the next stage might be to say, oh, well, now let's see if we can also use the same now approved drug or now in phase 3 drug for Parkinson's disease, or for, as an anti-cancer agent or something like that. And you get a plethora of different indications. And I often compare this to what's gone on in the oncology field with Keytruda, which is the checkpoint inhibitor antibody, where it started out and was approved first in melanoma, but because the fundamental biology was shared across so many different tumors, they were able to very rapidly do dozens of different clinical trials and get approval for each of these indications independently, until the FDA finally threw up their hands and said, all right, anywhere that there's PD-1, you can use Keytruda along with the tumor, which kind of gets us to the third phase of this natural history of our ideal drug, which would be some point where we stop thinking about the indications that are listed by the FDA, Parkinson's, cancer, kidney disease, diabetes, whatever, and start thinking about prophylaxis. How do we use a drug proactively, where we're treating something, but we're treating a molecular dysfunction, not a pathological dysfunction? And I wonder if that's something from maybe all three of you guys. I would just love to hear, maybe Tom, then Eric, then Bev, if that's a rubric that makes sense to you in this field. And then we can kind of use it to jump to the third part, which is how do we get to part 3 of the prophylaxis? Yeah.
So, I mean, I do think that that's probably the best way to go, given that we don't have ICD-10 codes for aging. There is one for frailty. There's one for some other things that have sort of emerged. They're still a bit nebulous, but there's really no endpoint that we can think of that's ready for prime time. And I don't mean to make this about our investors, but it's interesting when you speak with people who are potentially interested in making investment in your company. You sort of have people who fall into a couple of different buckets. I mean, one is, I want you to solve aging, go after it, and please do that in 5 years, right? And so there's that kind of I would call it almost an overly exuberant approach that is a little bit frightening, actually, if you think about what the expectations of that investor might be for short-term successes coming out of a program that are really not particularly, they're not precedent. There are other groups of investors who are only interested in understanding what your specific shots on goal are. How do you get to a value inflection so that you can either go public or they can get their money back from an M&A? And that, again, sort of focuses you on individual indications. And so what you find is the one group will often, if you talk, say, for example, about autosomal dominant polycystic kidney disease, which is a beautiful setup for mTORC1, they get very nervous when you start talking about, and this could be useful for even diabetic nephropathy, which is a small step in many ways from ADPKD relative to the whole aging sort of spectrum. So I think this is a conversation problem as much as it is a logic problem. And so I think the clearest path will be through those sort of, as we call them at our company, gateway indications. You could think of as beachhead indications. But if there is a way to define a development path that allows you to sort of validate the compound, the tissue, the dose that you're using as well, and then to expand that indication reach at the time that's right, I think that becomes a very actionable type of an approach. But these are actually really, really challenging to find. One, I think, needs to be very thoughtful about how to sort of take those steps as you move along. And this is again, I think, part of the conversation we really need to have more broadly. You mentioned the checkpoint stop. So, okay, what do we know about checkpoint? Well, first of all, the idea is at least 15, 20 years old. The approach has been fueled by the entire pharmaceutical industry going after it with massive advocacy, life-threatening illness, and a very clear sort of molecular understanding of exactly what's going on in the cell type that you're trying to treat. And so, I think this is both an awareness problem and an advocacy issue that we have to really take on at the same time, because the idea that we would be able to generalize treatment for something as broad as mTORC1 hyperactivation because you're getting old and that's everybody, or maybe you've eaten too much protein your whole life or something. But I think the problem is really very large, and I do think it comes back to this advocacy question as much as anything else. And for those of us who are in the field, I think the more that we can kind of align our messaging on these concepts and to sort of develop a convention, if you will, about how to think about these indications and how they might advance, this is going to be quite important, and keep the hype out of it as much as is manageable. And so, again, I apologize. That's a little bit free-form thinking, but I don't know if I answered your question at all. But I think that's kind of what, yeah.
Yeah, very good seeds for the discussion. It makes me, as we think about the bridge to this, to this bigger picture that you alluded to, Tom, I'm just really curious, quick reactions from the three panelists. How important do you think that a clinical trial that could start sooner for that gives both a hard endpoint and some biomarker-based feedback, like the TAME trial for metformin, how important do you think a trial like this, and it doesn't necessarily have to be with metformin, you could imagine one for other generic drugs as well. How important is a trial like that going to be for the development of this third bridge in the longevity space? Bev, could we maybe start with you in that one, and then Eric, and then hear from Tom again?
Yeah, sure. I mean, hugely important. I think part of what you have to do is influence the regulators, and a little bit like where the cell and gene therapy sector was 10 years ago, give them the confidence to break with what they may naturally have done and naturally have looked for in the past. So absolutely. I'll keep it brief because I know we've probably got much more to discuss.
Yeah, Eric, how do you feel about the TAME trial? Oh, you're muted still.
Thanks very much. Yeah, so I think that, yeah, it's really important to work on this from both ends. So I think, as sort of, a biotech company pursuing a specific target, we're trying to kind of start with sort of acute short-term things and expand to the full potential ultimately with adding multiple indications, and adding inspiration around, this is really going to affect health span in a broad way. This will be relevant to multiple indications. It'll ultimately be preventative. But, with the TAME trial, you're kind of starting at the other end and trying to get regulators and the public comfortable around this idea that, yeah, that end goal is something worth going after, where you're just, really frankly targeting multimorbidity. And so I think it's really important to build confidence and familiarity around that so that, we're going to have then, an easier time getting to that end stage too. So you're building from both ends, and I think that's important.
Interesting. Tom, what do you think?
Yeah, I'd agree. I think the TAME trial is absolutely a wonderful kind of first balloon to float in this area, and metformin is just the perfect drug for it. The question will be like, did they get the dose right? Are they measuring the right things, and are the impacts what we're really hoping for? But I think it's a really wonderful and important step to go forward with. I don't mean to sort of direct this toward COVID, but one of the things I think we're all seeing with COVID, for example, is this very, very clear link between chronological age and morbidity, mortality as well. and that the same issues that are most closely associated with age-related morbidity are also the comorbidities that drive mortality and morbidity with COVID as well. And so I think, as a field, we actually have a very live experiment running right now that we shouldn't miss the opportunity to capture the learnings that come out of this for our space. And so, for example, I'm just curious to know, is anybody mining whether patients who are taking metformin are faring any better than those that aren't, assuming that they're all diabetic, right? So high-risk population on or off metformin, most should be on, but not all. Is there a message that can be derived from that? So groups like Optum or other like large medical record mining groups be able to tease that out in time. And so these are the sorts of things that I think we should really leverage, because the entire population of the Earth is tuned into this at the moment, and we're watching an aging experiment play out right now. And whether we wanted it or not, we have it. Yeah,
exactly. And in speaking with Nir Barzilai, who's leading the TAME effort, I know that that's very much on his mind right now. And as TAME looks to be, raising philanthropic money to get underway as soon as possible, I know that one of the things that he's added in is a look at, in a placebo-controlled way, as we experience a second and potentially third wave of COVID infections, they actually want to be measuring whether they can see a measurable effect in both the infection rate and the response to infection, how severe it becomes in patients, whether someone is on or off the drug. Because, I think, looking back at all of this, we will probably observe that if something like this COVID pandemic comes up again, we want as few people as possible to have the immune systems of an 85-year-old.
Right. Yeah.
Okay.
Absolutely. And I just, are we getting back to your main question there, James?
We are going to go back, but yeah, Eric, please go ahead.
Oh, no, that was, I just wanted to know if that was the right time to continue on that main question. Yeah.
Yeah. So
how to expand. Mm-hmm.
So yeah, back to the main kind of final topic here that I want to touch upon about going broader. But if you want to jump in.
Oh yeah, no, just about the going broader actually. So that was the question you had already asked. And,
exactly.
I just wanted to jump in because I think there's a way to think about this which is really interesting. The idea you're starting with one indication, you're going broader, but that's only one part of it. And you already alluded to this with the Keytruda example and other things, that ultimately you want to take a program and expand it in sort of three ways. And one is indication space. So, building evidence around multiple different indications that can ultimately kind of span the healthcare experience of the average elderly person. So if you build confidence around enough indications, really that's like, you might be treating one thing that they have, one disease like heart disease or kidney disease, but then also simultaneously preventing their other diseases. So that's one key way to expand. The second is in temporal space. So, in terms of the progression of disease, maybe you start out treating disease, but then you end up, treating, early stage disease or pre-morbid disease, and then ultimately are preventing disease. So that's another way. And then third is in kind of molecular space, which is where, if you understand the mechanism of your target and your therapy, that you start out just, targeting, one indication that is relevant to that mechanism, but ultimately, build confidence around the fact that, there's a molecular signature that can be used to identify people who will benefit, maybe regardless of what indication they present with. And these three things are exactly the kind of the framework that we use to think about how we're planning our current clinical, stage programs. And, in the molecular space, of course, targets come from molecular data in our cohorts, and we're sort of also thinking about establishing those signatures, who's going to benefit. And also, as we talk about collecting biomarkers around, confidence that this will affect aging broadly around a broad range of indications. But anyway, just that rubric. So, indication space, temporal space, and molecular space. And I think that just synthesizes some things other people were talking about too.
I really like that framework, Eric. Recognizing that we've been having a lively and good discussion here, I want to start moving towards wrapping it up a little bit. But there's one piece that I think this discussion would not be complete without touching on a little bit more deeply, because it's come up half a dozen times already, which is, let's spend 5 minutes just talking a little bit about biomarkers in this field and what they mean, right? We've touched on them in the context of clinical trials, in the context of TAME, in the context of finding targets. So maybe, Bev, I would like to start with you on this one, since you brought it up as well. Like, from a tactical standpoint, if you're a small company in the longevity field, an academic spinning something out, someone that's wanting to collaborate with you, how would you recommend that a new group think about biomarkers? What are the most important things that we can do in the biomarker field over the next 3 to 5 years that you want to be helping folks with?
Okay, big question. What can we be doing? We can be making it easy to access resources that have been collectively sourced, which go across countries and perhaps further, and look at how biomarkers extrapolate not just for target validation, but how they can be used as almost companion diagnostics, which has been done in other fields. And as an academic group spinning out, making sure that they are signposted into how to use that and how to use it well. I think that's probably one of the single biggest things that will help them.
I like that. I think there's an enormous, and this is something I would love Eric to weigh in on, as someone, because Eric's both a physician and a data scientist, which is, I think, one of the cool things, and probably why he has his position in BioAge, or a part of why you have your position in BioAge, because for so many of my colleagues, understanding both the scientific and medical elements and the enormous just avalanches of data that we've been generating in the past decade or two, figuring out how to bring those two things together is a big part of what has stopped us from moving further than we have, I think. So maybe, Eric, I would just love your thoughts. How do we wade through the avalanche of data to find something useful in this biomarker world, and how is you mentioned a couple things where BioAge is using it on the target selection standpoint, but I'm curious how BioAge thinks about it for the companion diagnostic piece that Bev just touched on, as well as the surrogate endpoint piece that we touched on earlier.
Sure. Yeah. So that's a number of points. So remind me if I don't get to all of them, or prompt me if I don't immediately get to all of them.
It was a rambly question, but let's just hear your perspective on the overall space,
sure.
Perhaps.
So in terms of wading through the reams of data, I think the first point is that what's always the most important in using a data-driven approach to address a question is starting off with the best, highest quality, most relevant data. And so honestly, it's, sometimes that's finding the right data that's out there, and sometimes that's creating the data sets that you need. And that's, that's basically the thesis for the genesis of BioAge, that we wanted to create, the right human data sets by finding these, really high impact human aging cohorts where you had people who'd already been tracked for many decades, starting healthy in middle age. And you could have, this, track this health span of like a healthy middle-aged person and differentiate on a molecular basis between the ones who, went on to essentially age quickly, get, highly morbid, and the ones who aged well and were robust and healthy in old age. And these banked samples, we could then interrogate them with modern omic technologies and look at the whole proteome, the whole transcriptome, et cetera. And we needed to create those data sets because they didn't exist. And so that's one answer to your question, which maybe sidesteps it a little bit. But you want to start with the highest quality data, but you can also, of course, synthesize the data that's already out there, and you just need to take a practical approach, finding among all that data what is the most relevant, and then combining it, in a relevant way. And that's also, a huge part of our platform. It's kind of hard to to quickly summarize that approach, but it's essentially, it does get back very much to relevance, and then having the right methods and approaches to synthesizing those different kinds of data. So that's, let's say that's wading through all the data. And then you also mentioned, what was it, biomarkers, companion diagnostics. Yeah, if you want to build confidence that you have a molecular biomarker that really says something important about aging, I think it does have to be human aging. Like, you can get some value from mouse aging or worm aging, but a lot less, right? So really, you have to start with this, this nidus that this is really something important for humans, at least in an observational way, and that can be done with an observational cohort. And then you can bolster that by sort of, does this biomarker change in people over time in an interventional way? And can you move that, with the therapeutics, and does that result in a benefit on clinical outcomes? So it's that sort of that triumvirate of things you want to establish to build confidence around a biomarker, and I think that's where we're going, leading forward. And as to where companion diagnostics come from, well, for us, they start with these cohorts where we do the target discovery. If you're discovering the targets from these cohorts, you also understand, what are the molecular changes in a broad sense that you might want to track as companion diagnostics. And then you can build confidence that those might work in animal models, but then ultimately you build that in your human trials. And I think that's the way that it's been done in other areas too, companion diagnostics for cancer and other sort of, molecular targeted therapeutics and for different diseases.
Awesome. Thanks, Eric. And then, Tom, this is going to, I guess, be one of the penultimate contributions to the panel as we're moving to wrap up a little bit. But I would just love your thoughts, kind of given everything that we've discussed today, as you start moving some of your drugs into the clinic for, first indication, then second wave, and then third wave. How do you imagine, like, what populations do you ultimately want to be treating? And can you give us and our listeners kind of the very rough sort of, I always hate asking for time frames, although it's always the first thing on people's minds, but sort of the cascade of events as you see it, and paint a little bit of a picture of like the endpoint for TORC1 targeted drugs. Is it just treating different diseases of the kidney? Is it treating people more broadly? How long is it going to take to get to the vision where you want it to be?
Yeah, so sort of one end state that I have thought of quite a bit is, look at the 60, 70-year-old patient with type 2 diabetes, probably also NASH, some pressure on the kidney for reduced kidney function. They're hypertensive, they have cognitive issues, they may be depressed as well. And, this is, and developing, clearly on their path to coronary artery disease and vascular stiffening, the rest of it. This is a patient who, in my ideal world, would not develop those concerns because they've already been treated with the agent that we've begun to work on. There's a lot to do between now and then, and I dare say that probably won't be alive to benefit from such a treatment myself, but I think that would be a wonderful state to get to. And so, I think the practicality where we are right now is that we need to identify endpoints that can be used for the practical matter of developing the drug, selecting the patient to be treated, and then monitoring the efficacy of the treatment. And if you sort of think back to those, like the sort of landmark biomarkers that have been used in the past, HbA1c for diabetes was developed as a way of linking glucose to diabetic retinopathy. Everyone bought that once it was shown to be effective. And it is not just the endpoint that you use to track efficacy of your drugs. It has nothing to do with hemoglobin itself, but they're used. It's used as an endpoint both for patient selection, treatment objective, changing course of treatment, and monitoring success as well. Another great one is LDL itself, of course, but in that case, it's a population biomarker that you're using to really reduce atherosclerotic risk in the population in a lot of ways. Somewhere in the middle of those is, I think, something that we can really begin to work with. So what would be really fantastic is if we could, in the course of the next 5-10 years, identify markers that are circulating or otherwise accessible in patients. We know that they track with aging as a chronological issue, but also as a morbidity issue, and where we could say without question that changing the level of that marker is a clear indication of benefit, and that one could select patients on the basis of that and treat to that goal. I think these are things that are really, really objectively obtainable if we have the right data. And these data must be there. They must be accessible. I think the BioAge program could be really exciting for that. But for me, that's it. And in the meantime, I think we have to do, what's practical. And, we're sitting, we have a tiger by the tail with the, so to speak, with the mTORC1 inhibition approach. The question is, how do we develop those responsibly and open up as much of those longer term opportunities as possible, while keeping an eye on the ball of how do we finance our company, how do we demonstrate the safety and efficacy of the drug, et cetera. So all of this is just, it's a long campaign that we have to take on. Yeah.
Yeah. Hopefully an exciting one. It's certainly part of the joy that I get, especially from my position as kind of an investor and builder of multiple different things in this space, where the way we kind of think about the difference between these two goals, kind of the eye on the ball, moving things into the clinic. We have dedicated teams for kind of running those things forward. Well, there's different folks focused on the public-private partnerships, the advocacy, the big long-term regulatory things. And one of the things that I love is that we have been able to kind of like dedicate resources to that, because there's a certain economy of scale to many of these conversations, right? Like the challenges that BioAge and Navitor and half of my portfolio companies face are kind of the same. So as this, the same in the long term, vastly different in the short term on the investor level. And so something that I'm very much looking forward to as this whole field matures in a way is for groups like ours to figure out better ways of working together to achieve some of these advocacy ends, because I think that we have an opportunity where a rising tide in this will really lift all ships. It's just a question of how do we combine the right resources to hit those goals on the regulatory side, on the data collection side, and so on. So this is going to be a fascinating thing for, I think, all of us to be a part of over the next 3-4 years. Great. Well, yeah. Bev, Tom, Eric, thanks so much for spending some time with me. This was a really, really fun panel discussion. I know we went a little bit longer than expected, but hopefully our listeners won't mind too much. And I hope that all of you enjoyed it, all of you listening enjoyed it, and that we get to talk more very soon.